Good afternoon and welcome to this month's Dean's Research Seminar. I'm Professor John Fazakli and I'm the Dean of the Faculty of Veterinary and Agricultural Sciences here at the University of Melbourne. And I'd like to start by acknowledging the traditional owners of the land that I'm on today, as well as the traditional owners of the land that you're situated on, and acknowledge that Indigenous Australians have been custodians of the lands and waterways of Australia for thousands of years. I pay my respects to their elders, past and present, and acknowledge the role of Indigenous knowledge in our academy. Professor Michael Tausz from the School of Agriculture and Food will present today's Dean's Seminar, which is Adapting to Climate Change and Drought, Are Stress-Tolerant Plants the Right Goal? Professor Tausz completed his PhD in plant pathology at the University of Graz in Austria in 1997. He was a Fulbright Scholar at the Forest Research Station in Riverside, California in 1999, and in 2003 took a research position at the Forest Science Centre for the Victorian Department of Sustainability and the Environment. He joined Melbourne University in 2004, where he progressed to full professor and held roles including head of the School of Forest and Ecosystem Science, Associate Dean for Research Training, and he was also the University of Melbourne lead on the Australian Grains Free Air Carbon Dioxide Enrichment Programme. After short stints at the University of Birmingham in the UK and then Central Queensland University back here in Australia, Michael rejoined the University of Melbourne earlier this year as director of the Victoria Drought Hub. This is one of 8 drought hubs set up across Australia by the federal government. Michael is a highly cited researcher with an H index of 35, and he's published more than 200 peer-reviewed publications. His specific areas of interest include plant biology, tree and crop physiology, and environmental plant science. Michael, over to you.
Okay, thank you very much, John. Thanks for the introduction. And I start to share my screen to jump into the presentation. So, hello to all, and thank you very much for giving me the chance for another introduction. As John already mentioned, I have a history with University of Melbourne, and for giving you a little bit of background, what I did then and maybe what I did on my vacations when I was not at University of Melbourne, I start with few slides there. John already mentioned the Australian Grains Free Air Carbon Dioxide Enrichment Program that ran for quite a few years between Agriculture Victoria and the University of Melbourne. And I think it's still one of the very good innovative experiments that were run in Australian crop science. And that's what I did during my final years at the University of Melbourne. But it also gave me, apart from good papers and good results, also a bit of an understanding that in these times where climate is changing so quickly, we cannot use traditional methods to adapt our agricultural practices or say select for the new varieties of crops that deal with the future climate, because we don't have the future climate yet. And this installation, free air carbon dioxide enrichment, that added carbon dioxide to the atmosphere, because we know that carbon dioxide is going up in the atmosphere. And that way we can look sort of into the future and understand better what type of crops and what type of practices we might need in these quickly changing times. So following on from that, when I left University of Melbourne, I went to Birmingham in the UK to a position as director of the Birmingham Institute of Forest Research. And that was that is a pan-university interdisciplinary institute about forests, but it is also built about one of these big experiments. And it is like the AgFace experiment, an experiment that exposes a whole mature forest canopy to elevated CO2 to see how forests deal in the future with these environmental conditions. During that time, I also learned that such marquee experiments are pretty good vehicle for engagement with the public or with industries to get our scientific results and our scientific cutting-edge research across to where it has impact. And that forest was called by a BBC journalist the sci-fi forest. And that's why it's such an installation can be very attractive for the general public. And here you see a scientist in blue explaining the experiment to some local here. I was always interested in the relevance of the research and in having real-world impact, because there is always quite an opinion that scientists don't want to go out and communicate, and that research results are not taken up and don't have that real-world impact that the really good cutting-edge results should have. And so that's why I was attracted to the position as director of the Victoria Drought Hub, because the drought hubs in a program by the Australian government, the Future Drought Fund, they are doing exactly that. They're designed to do exactly that. So the Victoria Hub is a consortium of 4 research universities, Agriculture Victoria, and 5 farming organizations, local organizations that deal with engagement, innovation in a regional sense. So this is exactly designed to make research impactful. The Drought Hub, the Victoria Drought Hub covers Victoria, headquartered in Dookie at the University of Melbourne, and has nodes all over Victoria run by local engaged organizations, the partners that we have there. And the design, and this is sort of my own words now, is to improve that pipeline from research, development, extension to adoption and commercial outcomes for society, for the regions, and for agriculture. And that is done by a lot of communication and co-design activities to link that pipeline up much better. So I pause here. This was just to give you a bit of a background where I came from and what I'm doing here at Melbourne, and I'm very happy to talk about any of that. But this is a research seminar, so I had to think about good research topic. And that's when I made up the title, and then that got me really thinking what my answer to that actually is. So we're looking into the future, and the climate change predictions talk a lot about stress conditions. We talk a lot about drought, of course, in the Drought Hub. And even if we have now maybe even 3 very good seasons, the next dry season will come certainly, even without climate change, because Australian climate always was already very variable and prone to extremes. So do we need stress-tolerant plants? Do we need drought-tolerant plants? It seems to be a no-brainer. And if we look at, this is wheat, one of the main crops, and we'll talk a little bit about wheat because there were previous seminars talking about wheat. And wheat is growing quite often under dryland conditions, and it needs to be drought tolerant to some extent. But do we want real drought tolerance in plants? And this is a bit of a provocation now, and it's even not my invention, but I unfortunately forgot who said that originally. I quite like that, because the term is, if we are really successful with creating the drought tolerant crop, we end up with a cactus, and we already have that. So that's just a really nice illustration that there is a very strong trade-off between tolerance traits and growth and yield traits. And so we can't just look at which plants are drought tolerant and try to put all these traits into our crops, because that won't work. We will end up with a cactus. They are very good at surviving drought, but they won't feed us, even if you like dragon fruits or cactus figs. So we, of course, in our staple crops, these are annuals. They're not drought tolerant plants. These are weeds ecologically. So there is a dynamic to their growth, and a dynamic between different phases, from vegetative growth. Tillering means stem formation in cereal crops. I come back to that example. And then, of course, into reproductive development, because we eat the mature seeds. So it wouldn't help us if that crop was very drought tolerant, but doesn't produce seeds. What we want is that they go into a lot of seed productions under difficult conditions. And Australia is quite unique. And in a previous seminar by James Hunt, he emphasized how Australian growing conditions are, for wheat, for example, can be quite different from most of the other places where they grow wheat. So there is a lot special about that as well. Now, when I did a bit of research for that seminar, a bit of reading, I saw with interest that there is work on tillering, on stem formation, because we've done some work on that before. But the basis for that is that cereal yield is determined by the number of heads, the number of, not only, but also by the number of heads. And that's determined by how many tillers are formed. And so tiller formation is repressed when there are stress conditions or unfavorable conditions. And so these guys here, published just a year ago or so in Science, a really high-level publishing, they work on explaining this tiller suppression in wheat, in rice, and try to get rid of it. And the logic is that if these low nitrogen conditions here do not cause a repression of tillers, then plants may grow as well under low nitrogen as they do under high nitrogen. I found it quite interesting, and we certainly will learn a lot about how tillers are formed, but we have to also take a step back and see what does it mean in practice. Because under Australian conditions that are very different from rice growing conditions, of course I admit that, the relationship between tillering and yield at the end can be very different. And Australian scientists have long already pointed out that high tiller formation in wheat, for example, can be an unwanted trait. Because with this seasonal drought that I pointed out before, the crops then mature and fill their grains under increasingly dry conditions. So if you have way too much tiller formation at the beginning, they may run out of water very quickly. And a phenomenon that was called or is called haying off may eventuate, that you have lots of hay produced, but hardly any grains. And that's, of course, not what we want. So quite in contrast to this simple logic to say, well, if you have more tillers, then you have more yield. If we look at the dynamic, the growth dynamic of actually the crop in the field, we come to a different conclusion. And that's, of course, has been work done by CSIRO for a long time, and they produced, for example, a trait for breeding, a so-called reduced tillering trait, a trait where those varieties formed fewer tillers to start with. And they called it TIN, a tiller inhibition gene. And it was tested a lot in Australian field conditions. And this is from work that one of my ex-PhD students did with CSIRO then, characterizing lots of field data, comparing varieties side by side with and without this trait. And the way it's shown here already shows, illustrates that we don't want to have drought tolerance. We want to have water use efficiency. So water supply on the x-axis against grain yield shows us water use efficiency in the slope of that maximum line, or so-called boundary line. Now, this is also long known, originally in a paper by French and Schultz, that shows what the limitations under Australian growing conditions are. And all these points are the real points from the field, but none of the points is over here. So this tiller inhibition gene didn't actually change water use efficiency. In fact, if you look at the very low end here, it's even, you would say, the line that characterizes this water use efficiency is even worse than without that gene. But what happens at very, very low water supply? They would still yield whereby without that tiller inhibition, you wouldn't have yield, you would have hail. So we did some work on that under high CO2 as well, just to make sure that we can try to look into the future how such traits would behave. And this is done through modeling, using experimental results, but then putting those to calibrate crop growth models. In Australia, the model APSIM is developed also by CSIRO, is of course the standard crop growth model. It is sort of limited in interpreting the real physiology of plants, but it is very practical. And if you do that, you can see that this gene, this trait, would have an advantage in all the areas where it's green, or would have a disadvantage where it's orange or red. And that's over a long period of historic growing seasons, you would end up with an advantage or with a disadvantage. So that could give you a bit of a decision whether or not to use this trait. But with our experiments, for example, from the egg phase experiment and with future scenarios that the Bureau of Meteorology and CSIRO produce with climate scenarios, we can also try to look into the future and say, is it really worth looking, say, 20 years ahead to have such a trait? And this is a bit hard to see in this comparison, but there is more green in this future scenario. Want to have more details? We just published a paper not too long ago in Global Change Biology. So I used it in this talk, not so much about that specific trait, because it's been around and studied for a long time, but just to illustrate, first of all, how important it is to design crops to the dynamic growing conditions, but then also how difficult it is to come up with a trait that breeders could use that is clearly beneficial without having a disadvantage anywhere. Because this, again, is the comparison between crops with this new trait and without, and they're grown side by side, and the dotted line would be the one-to-one line, if they would yield the same having the trait or not. And the actual line, the fat line here, is a little bit different from that, and it shows that at higher yielding conditions, the trait is a disadvantage. And that's now a problem because it's an advantage at low yielding conditions, disadvantage at high yielding conditions. And the real conditions, say 4 years, might be like that. Here, 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 because we have such a variability in growing climates. So how do we decide? There is a risk there, right? I take a bit of a step back there and say that this is not only looking at something that we've done now in the last 10 years or so, but it is still the holy grail for breeding research in some ways. And I took this citation because it comes from the Wheat Yield International Wheat Yield Research Consortium. And they still say that understanding better this growth and defense, growth and adaptation trade-off is really important to target future breeding. Now, we have this environmental variability within growing seasons, and we know that, of course, for a long time, and wheat cereal researchers, breeders account very well for that and can target traits to line up very well with the variable within season growing environment. Between growing seasons, it's already a bit more difficult because if you plant a variety, plant it and that's it for the season. So there is a risk there that needs to be evaluated. But now we go into a new phase with the new climate where shorter term variability comes into play even stronger. And that's because all the predictions for climate change into the future, where they really all agree, is that we will have more and more extreme events because global warming, the atmosphere is warmer, there's more energy in the atmosphere, and it makes the weather more extreme. So we will deal with more heat waves, more short and sharp dry spells in terms of drought, possibly unseasonal frost events, because everything becomes more extreme. And if we have that sort of environment, then we go back to the survival question again, because then it may be important for crops to get through that, rather than having a dynamic adaptation to the whole growing season. So you might have, during vegetative growth, a short and sharp heat wave, droughts that causes drought stress if there isn't enough water, or is just heat stress. And then if the crops are damaged here, they would have trouble to realize all that potential here, where there may not be no stress anymore. This is a real-life example from CIMMYT. That's the International Wheat and Maize Breeding Center in Mexico. And these are heat damage symptoms on wheat leaves. And you can see that they are just where the leaf was most exposed to the sun. Now, that's not directly because it was warmer there. That's because direct damage on plant tissues, on photosynthetic tissues, generally starts at what we call photooxidative damage. It starts in the mechanism is because plants have their plants do photosynthesis. And that's actually a pretty tricky process to regulate. And if it's not well regulated, and if there are stress conditions, the light that plants absorb to run photosynthesis is likely to do damage. And we have a situation called photooxidative damage. And plants, of course, have evolved protective systems to shield them from the immediate effects of that. But it depends on how strong they can shield themselves and how strong the stress actually is. But you have heard about compounds called antioxidants. People will try to sell it to you as quality in your food and so on, because we also need these protective compounds. But plants make them in response or in preparation for stress. So photooxidative damage comes from the light that runs actually plant lives in the first place. So maybe back to the questions. Do we need stress tolerant crops? I tried to say first, not fully tolerant, but adapted to the dynamic conditions. If I looked now into the web of knowledge, there are about 8,000 papers in there with crop and stress tolerance. So obviously people think it is important. And there are about 2,000 that say crop, stress tolerance, and antioxidants, which are these chemicals that are protective against photooxidative stress. I obviously didn't read all 2,000 or not even did a thorough review, but I looked at a few, and many still follow a very simple and, in my opinion, quite flawed logic here. We know that plants under stress have higher levels of these defense compounds. So the logic is higher defense compounds confer stress tolerance. I never quite liked that, and that brings me back to work I've done quite a while ago, and actually on drought stress in apple trees. In one experiment, we measured defense compounds. At the time, that was quite tricky because you could only measure single compounds with complicated methods. So most people didn't do any timelines or dynamic responses. And that was, I believe, the first paper where we did such things. But it also showed us that the plant stress response, even at that biochemical level, is dynamic. So stress in the drought experiment increases in intensity, and then you have different changes in these antioxidants. Black, and sorry for putting that together from different sources, but black are the stressed leaves. It's leaves of trees and not the fruits as shown here. And white are just the controls. And you see in the controls, there's no change in any of these compounds, but in the stressed ones, yes, they build up these antioxidants. You know, ascorbic acid, it's not important to remember that now, but vitamin C may be something everybody knows as a defense compound. Glutathione is another one that's very important in plants as well. So yes, if plants respond to stress, they seem to have more antioxidants. Does that mean they are more stress tolerant? From that, we can't really say. But what we can say is that there is a dynamic to that response from an initial stage. And nowadays, we could characterize it much better with metabolomic methods where we can measure lots of metabolites at once, not one by one as we did at the time. Then we have an active defense phase. But we also can see that there is a breakdown of the system, and that's before we see anything at the plant themselves. So these stress responses are very dynamic. They could be helpful if we look at them in crops as well, but we can't, it's very difficult to select for it because it's so dynamic and we never know when we look. But there is progress made because environments generally are always dynamic. And we can't just say we have a tolerant plant. We have to see how plants perform in this environment and when they engage defense and when they use everything for growth. Growing environments are generally dynamic. And I come back even further to something I did in forest sciences when we were studying the canopies of these nice, cool temperate rainforest, nothofagus southern beech trees. And we were interested in how the competition between lower canopy, higher canopy leaves works out and what determines these competitions. And the dynamics come in when you think about the light environments these leaves in the canopies are exposed to. So in this graph, on the vertical axis, that's light intensity as it is experienced by leaves further up and lower down in the canopy. Further up, lower down. And you see these curves don't really look like what you would expect from a sunny day. And that's because many times during the day, the leaves are shaded by neighbors, by other leaves, and even more so when they're further down the canopy. So in fact, instead of having sunrise, it goes to the highest light and going down again, you have shade, 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 then suddenly very high light, back to shade, highlight, back to shade. And photosynthesis is directly light dependent. So production is directly light dependent. And you would calculate in simple models production just from light. And what we did here is we actually investigated how much error that would cause if we do it that way. We looked at this shade, light, transition phases. And we looked at photosynthesis, how it kicks in, but there is actually always a delay until it's fully up and running. And that also happens with stomata, which would be important for drought stress. I'm not going further there today. Another interesting thing that happens here is that plants engage protection when there's high light. So they become actually less efficient in using the light for photosynthesis because they don't have to. That is shown we can measure as photosynthetic efficiency. We can measure that with fluorescence methods or different methods these days. And again, that goes down when light goes up, but there is a delay to that. We did calculate that I can't remember the exact number, but about 20% potential loss compared to what it would be if plants could immediately react to the high light every time. There is another aspect to that, which I was always very interested in. This engaging of photoprotection is actually at the cost of productivity, of course. But as long as it's under high light, it doesn't really come at the cost because there's enough light and plants don't need to be so efficient. So we looked in more detail how that worked, and it is related photoprotection. One particular system is related to protective pigments, a certain class of orange, yellow pigments that are very quickly converted into each other. And they go from a relaxed state to a protection engaged state back to a relaxed state. Why do I say back? Because in this experiment, we tried to see the shade to light transition. So you can see that biochemically we studied protection is engaged and efficiency is lost. And then we darkened the leaf and looked how long it took for that all to go back to fully relaxed state. And we did that to explain the competition within the canopies, but we did pick up that this relaxation may actually take quite different amounts of time in different positions and in different leaves. We didn't go further at the time, and to be honest, when I thought about it, I didn't really think it would be that significant for crop canopies, because most crop canopies, most production happens at the very top, where there wouldn't be that much shading. I think I was a bit wrong, because if I look, when I look now a bit in the literature, there is a very new article summarizing that exactly same effect for crop canopies. And this, I forgot what it is now, I think soybean or tobacco, like crops with maybe denser canopies, but leaves just underneath the top layer experience again this sort of light environment during the day, with lots of stop-start, rather than continuous possibility for production. And there is a delay in them fully starting. This is assimilation, photosynthesis, so that would be the food production, and that would be the ideal production. So there's a delay with every light fleck, and then there is also a delay in relaxing back to full efficiency, and that leads to very low production under lower light, because that's when you would need the efficiency. So both these effects, the engagement of photosynthesis and then the relaxation of the engaged photoprotection, come at a cost. And those guys calculated in another paper that it's about 20% loss of potential production in a typical crop canopy. And there was a quite nice tries with tobacco plants, where they engineered that photoprotective pigment metabolism in a way that the reaction was faster. And in that paper, and it's again published in Science, so I would not be one to argue with that, they showed that those engineered plants had 15% greater dry matter production and carbon fixation. I think it's very tricky to manipulate that, because this is an important protective mechanism. But if you only make it respond faster, both in up and down, maybe it doesn't really come at a trade-off. Now, the other way to look at that, rather than manipulating it and hoping it doesn't come for a trade-off, would be to see whether there's variability among existing crops. And that would be really a lot of work if we had to do it the way we did in the canopy, by taking repeated samples, doing biochemical analysis of each of the compounds. But there is another way to it. And if Pablo is watching now, I should have talked to him before I do that, but I used this graph quite a lot in lectures already, so I know it's already a while ago, but I think it illustrates that with the new methods that we have available, there may be another way of measuring the dynamic of these responses, because this image here shows an index, a reflectance index that is linked to this photoprotective pigment engagement and relaxation. And plants engage protection under additional stresses, which is also shown here, because this is from, I believe, a drought or watering experiment. So these are drought-stressed trees in an orchard. Here's the orchard. And the blue ones are well-watered ones. And you see this signature of protection engaged is where the trees were drought-stressed. But we could look at it possibly in a dynamic way. Are there any varieties in a field or spots in a field where this goes faster or slower, rather than whether it happens or stays like that? Now, as I said, I didn't even intend to give a lot of results or summarize all the research that I've done. I wanted to put out a few ideas and help myself to get ideas ready as well. So let's go to some what if. The first was already, and that was already shown, that this photoprotection may give us a way into select for more dynamic responses that may help, again, to boost crop production without really losing anything in stress protection. But if we go back to this dynamic stress protection example, which is from leaves in drought-stressed apple trees, what if we could find plants that respond quicker here, or that respond stronger here to the same stress? What most people do is they look for plants that have high level of protective substances. And you normally go out and sample that once, twice. But in my opinion, it will always come at the cost of a trade-off. So if you have plants that consistently have lots of defense compounds and invest a lot in defense, they probably will pay in terms of yield potential and yield under good conditions. And given their variability in conditions, that's not really what we want. But what if we could get plants to react faster and stronger here? Is there any chance to select for that? And possibly new methods in measuring these antioxidants will help, because with metabolomic methods, it's much easier to take repeated samples and to have a large number of metabolites at once. And so we could screen much greater numbers of plants. But there is even another interesting development, which came mainly from plant pathology, from responses to pathogen attacks to plants, because that's where actually that trade-off between growth and defense is much better studied compared to trade-offs with abiotic stresses. And this is my last my last slide, so I've made it really difficult. But it describes a process called priming. Can we prime plants to be more responsive or more better defended to subsequent stress? And there are good, I would say, good hints that it's possible, but we don't really know a lot about these processes yet. But the principle goes like this. Let's start at the full blue and red lines down here. And they show the defense response. You could think defense compounds or any defense response of plants invest in defense. So blue is just a normal plant. We don't do anything to it. It doesn't have any particularly engaged defenses, but then a stress hits it. You see here it says triggering stress. And with some delay, defenses are engaged, and they are not engaged to a terribly high level either. And then if that defense is overcome, there might even be a damage or decline, or the plants recover again. And if you look at the blue dashed line here, that's the cost. So there is no cost because the plant is not doing anything, but they lose, of course, a lot of potential that could be put into yields when they engage their defense and when they suffer damages to that stress. Now, priming means, and it was observed that moderate stress exposure can make plants more tolerant to subsequent stress. So if you have a priming stimulus, they call it here, you have a moderate response, but no real damage. And it goes back again. If you measure antioxidants and stuff, you can't see anything here really, or probably you have to see it somewhere, but it's not known yet. And the plant can go into a primed stage. They call it ready for action, ready for defense. That primed stage may be maintained for quite a while. And you see the cost here in the dashed red line. There must be a cost to the prime stage, but it may be very modest compared to the cost of suffering damage and then in the subsequent growth, if we think of our cereal. That priming does something to the plant. It's ready for action. So if now stress hits it, it reacts very quickly and very strongly with defense engagement. That has been shown for pathogen responses in several systems, not so much yet for abiotic stress. But this strong response would then mean that there is a big benefit in terms of overall costs to the plants because they don't suffer damage, they protect themselves, and then ideally they can even relax quicker. So this is a bit of a thinking exercise of what else we might be able, apart from what breeding research is doing great things anyway already, and we learn a lot about molecular mechanisms, how to use this knowledge to put it then into real plants, but what kind of things could we still target that hasn't been tapped so much yet? And I think the dynamics of plant and growth environments has been quite difficult to handle and still is, and we're just about getting tools that allow us maybe a bit more there. We cannot really overcome trade-offs between defense and growth and yield. That's just a principle. It's an evolution principle. But we can possibly tighten the time frames of these trade-offs and thereby help to go always more on the side of growth and yield whenever possible, but still have defenses ready whenever needed, and not have defenses ready just in case. And if they're not needed, you pay a high penalty in your growth and yield. So with more extreme events, this defense becomes more important because it's really important that plants may survive a short, sharp event. So maybe two questions that can lead to research questions. Are there plants that engage and relax defense mechanisms faster? Are there lines already existing? I think we don't know yet. We haven't really looked for that yet. And the other big question is, can we have some handle on priming plants for stress response? My thinking is heat waves are predicted, but plants don't know they're coming. Could we give them some sort of trigger that they might be better protected? And I'm very happy to discuss any of that because it also helps my own thinking where to go. Okay, I'm at the end of it, and thank you very much there.
Thank you, Michael. So if you've got questions, can you put them in the Q&A, please? And Michael, if you want to stop sharing, perhaps we can take questions. So questions in the Q&A. Let me start while colleagues may be thinking about some questions. So if I follow what you were saying there, Michael, if you had a transgenic plant and it made lots of additional antioxidants, which would be protective when a stress came along, that plant wouldn't grow as well. Or is that a prediction, or has people actually tested that and found that's the case?
Yeah, it is a principle, and I think it's true because if a plant invests more into defense, then it hasn't got that much to invest into the growth processes that it needs. Now, with antioxidants, it's a tricky thing because they are very well regulated within the plant as well. So there have been multiple attempts to try to produce plants with more antioxidants and show that they are more stress tolerant. But it is a whole network of defenses, and I would say the outcomes so far have been mixed. So even that point that they really would be more tolerant is not so easy to prove. So I think we need to link this understanding really up with more practical real world work. And I think these new screening methods and methods to measure things quicker, that could help us there. So yes, but in answer to that, yes, I'm saying these trade-offs are a principle and we can't easily overcome it. There is always saying that things don't come at all at the yield penalty, but under certain conditions, not when the defense is needed.
Well, another way to do it, of course, is not to rely on the plant to reduce the stress, but for the humans to reduce the stress and to put up sort of shading systems. So, you know, how effective are those? You sometimes see them in orchards, and but I don't think you see them that often in the big wheat fields. I suppose there's so much acreage there.
Well, shading is another trade-off because, of course, photosynthesis needs light. So, but light, if there is some other stress, may cause direct photooxidative damage. So, yes, when there's a stress exposure, shading may actually help to just limit that photooxidative stress. But then if you have too much shade, your whole production is less anyway because you have less photosynthetic production. Some plants are very strongly geared towards low light conditions, and even as crops, they have to be shaded. It used to be a tradition, for example, to shade coffee trees because they come from a lower light canopy and they were not as protected against high light. I think it's not state of the art anymore because the new varieties can deal with full light as well, but they would still be exposed if there is additional stress. Yes.
Okay, let's turn to our audience and have some questions from them. Saeed, would you like to ask your question? And let's see if your microphone works. Saeed, are you there?
Yeah, hello, can you hear me?
Yeah.
Yeah.
We can hear you.
Yeah.
Yeah.
Welcome, Michael. Excellent talk. My question is spoke in detail about the relationship between the number of tillers formation and yield, and you touched briefly on antioxidant formation as a result of stress. The question is, has there been enough work on the relationship between tiller formation stress and the nutritional value of the product, meaning wheat, grain, or any other produce?
I don't really know. I'm pretty sure there was a lot of work on tillers and the general grain yield and also quality in terms of protein. I'm not quite sure. Did you want
like, for example, protein content, starch content, fiber content in the wheat? We did some work on strawberry and we detected huge amount of antioxidant production under stress.
Yes.
But the question, how about protein, starch, fiber, fat content in these produces?
I think under stress, okay, in wheat under stress, generally you have less deposition of carbohydrates if it comes to the grain filling phase. And you have the paradox that, for example, grains maturing under stress like heat have actually higher protein content, which is a positive quality trait, but they have less yield. But I know that that's interesting to hear the work about strawberries, because we've done work on tomato, trying to expose them to stress to make them higher quality, because what's the thing called in tomato? Lycopene is a quality, it's an antioxidant, but also a quality trait. But the problem was, again, that trade-off. It's possible you have tomatoes with very high lycopene, but you have very low yields. So that trade-off is also something that needs to be economically evaluated there.
Thank you.
And let's go to Ian Clark. Ian, are you there? You can ask your question.
I need to, can you hear me?
Yep, we can hear you well.
Yeah, okay. So my question really is whether the defense mechanisms in the ancient grains are any better or worse than those in the sort of evolved grain varieties.
That's a good question. I don't know at all in for cereals. I know there are some genetic differences in the general level of antioxidants for some tree species. We didn't find that apple experiment that I showed we did on several apple varieties was very similar everywhere, although some of them were older varieties and others are newer ones. I can't answer that for grains at all. I don't know.
Okay.
Ian, thanks for your question.
Oh.
And next question is Gio.
Hi, Michael.
Hi, Gio.
Great talk. I have a curiosity. And first, I'm very interested to hear that some of the approaches study on crop are very similar to what's happening on animal and even in fish, because the problems are the same. And when it comes to heat stress and antioxidant, there are a lot of similarities. I was very interested in the concept you mentioned about priming, which is actually something I'm studying as well in fish. I was wondering how early or late these stress stimulus need to be provided to be effective, and if that can even be anticipated cross-generational. So if we can sort of stress the seed formation and then affect the following plants. Is this known or not yet?
I don't think the timescales are known, because I would be really interested in look at that. There is some work done on pathogens, like when they're primed and then look at the defense response to pathogens. And I have to think, I don't know of any work in cereals. They did things in tomato, and I think it was in the order of a week or maybe 2 weeks. The seed question is another really interesting one, because I know of work where they primed seeds with tocopherol, and that's an antioxidant, and there was a positive effect on germination and so on, but I don't know whether that work has ever been followed up on that. Again, sorry, I can't really answer the question, but they're all really
Thanks, Michael. Now, just curiosity, because I guess, for example, in fish and animals as well, there's a lot of work currently done on the early stress you can give to an animal in fish at egg stage and even broodstock, so the parents. And you can actually clearly affect the capacity of fish to cope with high temperature, for example, by exposing fish to high stress, high temperature at very early stage of development. So maybe there could be something happening there as well. Lot of interesting things to do and study. That's great.
Yeah, that's good.
Thanks, Gio. That's interesting. There, the parallels, isn't it, between animals and plants? And I was thinking that as well when you were talking about antioxidants. And let's go over to Snow. Snow Barlow, previous dean of this faculty. Snow, are you on the line? Yep.
Can you hear me?
Yep, perfectly well.
Very well. Hello, Snow.
Hello, Michael. Welcome back to Melbourne University, very belatedly, but it's excellent to have you back. Fascinating seminar and very thought-provoking. For an old stress physiologist, we won't dwell on the old part. You would be aware of a very recent paper, actually sort of from Nature, about a basic assumption in plant physiology that the relative humidity within plant leaves was 100%, which now there is a paper reporting that it is actually lower than that, and perhaps as low as 80%. Bearing in this in mind, does this raise the question of a sort of re-examination of the potential for stomatal control one way or the other as a potential tool in stress tolerance work in crop plants?
Well, I stumbled across this, and I think this is always a tricky thing, because it was an assumption and it's still very difficult to measure directly, and there are lots of assumptions there as well. I don't actually think in terms of transpiration it makes so much difference. You could recalculate basically all the gas exchange data to that, because the assumption could then be, okay, somewhere between 100 and 80. But on the broader question, I do believe that actually the time course, the dynamics of stomatal responses is another underexplored area there, where I think the rapidity with which plants respond with their stomatal movements, I think that could be explored more. I'm only aware of some damages make stomata sluggish, and plants actually suffer drought stress because they don't close stomata overnight properly and have, unproductive water loss. But the other way around, whether we can select for plants that are faster, I do think there is still something in there.
Okay, thanks for that. Snow, I see you've got another question. Would you like to ask it while you're on the line? Then I'll go to the Hafiz.
Is that me, John?
Yes, no, yeah. Please ask your other question while you're on the line, and then I'll move on.
Thank you, John. Michael, this is a left field question in relation to your seminar today, but given your, sort of great experience and knowledge in the area of forestry and elevated CO2, what those of us who are interested in this current emissions debate of CO2 sequestration by forests in Australia, and you would be aware of the parliamentary inquiry occurring at present of whether it's bogus or not. But there are estimates that are inherent in that work that a lot of that response is actually due to forest response, well, forest and shrub responses, but perennial plant response to the current levels of CO2, which are probably 25% higher than they were a century ago. Could you comment on that?
Yeah, okay. So that's going back to my work in Birmingham, where that
Yes,
experiment was actually put in place to test some of those things, because that so-called terrestrial carbon sink is in the moment in those global carbon exchange models. We know what's in the atmosphere. We know, because it's a physical-chemical thing, we know how that affects absorption into the oceans. But the absorption into the terrestrial ecosystems is actually calculated. And it's pretty high. Yes, you're right. There is a carbon sink there, which is not fully explained by our current models that builds up from the ground, like the models summarizing globally and building up from the ground don't quite meet there. And that's what they call the terrestrial sink. So there was a lot of concern about it, and that's why they put in that experiment in place, because temperate forests and of course tropical forests would be the main contributors there. Unfortunately, not so much Mediterranean forests, because their rates are not as high when they're active. But the English deciduous forests have actually very high rates during summer. And yes, they apparently, otherwise there would be a part of the CO2 missing that goes into that forest. And the concern is that that may become saturated, or is becoming saturated. And then the extrapolations may be even worse than they are now.
Thank you.
Okay. Thank you for the question, Snow. And the last question of the day comes from Hafiz, which I like this one as well. Hafiz, are you there? Yeah, go ahead.
Thanks, John. Thanks, Michael. Wonderful presentation. Excellent information about phytochemicals, vitamin C, and antioxidant. One of the question that I have, as per my knowledge, like during the stress physiology, like phytochemicals produce and convert from one form to another form, and many of the metabolites produced, and during the defense mechanism, plant use some of the metabolites that affect the quality of the fruits and vegetables, like, and the production of the fruits or vegetables. So do you think, like, is there any way, like, we can reverse those, we can produce those phytochemicals again, like by providing some of the better conditions to the plant, or like, because in the end of the day, the fruits and vegetables quality destroyed because of those phytochemicals when they overuse those metabolites. So do you think we can reverse them, or we can adopt any of the strategies that can actually help to reduce this stress, like at the stage of the production of the food?
Okay, so you are now referring to end products of oxidative pathways that are then affecting the quality, because they are something like bitter in taste or things like that, right?
Yes.
Yes. I don't really think you can, because they are there, because they're not easily reversible. So that would be for phenolics are often acceptors for oxidative pathways. I think the way to do that, and that's a really interesting question, is to go back to those detoxification pathways that don't have a persistent end product. So probably if you have more pathways protecting through the water-water cycle that basically let electrons in circles back to water, over those that run those electrons onto persistent compounds, maybe that would be a way to look. But this is totally from the top of my head, and it's a really interesting question.
Thanks, Michael, because I'm just thinking like during the different type of stress, for example, water stress, heat stress, like different stress, they generate different type of phytochemicals. And then it definitely affects the quality of the fruits and vegetables in a different way. So if there is any way, like they can stop the pathways, or different, even like they can move from one pathway to another pathway and they link them. So maybe ultimately they can improve the quality, or maybe just better the quality at least, because that's what I was just thinking.
Yeah.
Okay, good thoughts. Nice discussion, but I'm going to bring it to a close there, and thank everybody for their questions, and thank Michael in particular for his presentation, which I found very interesting and stimulating. And Michael, you said you were hoping to marshal your own thoughts, and hopefully it's given you some time to think and some ideas as well. I'd like to thank everyone for taking the time to attend today's presentation, and I hope you'll join us on the 13th of September for next month's Dean's Research Seminar, which will be Professor Peter Mansell, who will present his seminar towards a sustainable Australian dairy industry. I hope to see you all then. Thank you.